The overall goal of the following experiment is to transplant enriched photoreceptor precursor cells into a recipient mouse retina. This is achieved by first isolating the retinae of young donor animals. In the second step, the postnatal photoreceptor precursor cells are isolated from the retinal cell population by CD73-based magnetic-associated cell sorting. Next, the photoreceptor precursors are transplanted into the subretinal space of recipient animals to facilitate their integration into the recipient retina. Ultimately, the integration potential and behavior of the transplanted photoreceptor precursor cells can be analyzed by immunofluorescence microscopy.
This method can help to answer key questions in the field of retinal regeneration, such as what is the capacity of transplanted photoreceptors to integrate in the neural circuitry of the host for restoration of vision.
The implications of this technique extend beyond the treatment of retinal degenerative diseases, once the isolation and enrichment of photoreceptor precursors by MACS can easily adapt it to GMP conditions.
Visual demonstration of this method is critical as the transplantation steps are difficult to learn. The subtle handling and placement of the needle in the subretinal space is crucial to ensure maximum success and reproducibility.
To dissociate the retinas, begin by briefly rinsing the heads of postnatal day 4 pups in 70% ethanol, followed by a PBS wash. Then transfer the heads to cold HBSS. To enucleate the eyes, for each eye, first open the eyelid. Then, using a pair of curved forceps, grasp the organ at the optic nerve region and pull the eye carefully out of the orbit. Next, to isolate the retina, introduce the tip of a pair of closed scissors into the optic nerve and open the blades. Peel the retinal pigment epithelium choroid out and use curved forceps to remove the lens and blood vessels. Incubate the isolated retinas in a 1.5 milliliter reaction tube containing papain solution for 30 to 60 minutes in a shaker at 400 rpm and 37 degrees Celsius. While the retinas are incubating, dispense 1 milliliter of ovomucoid solution into a 15 milliliter reaction tube labeled 1. Then, to a 2 milliliter reaction tube labeled 2, add 60 microliters of DNase-1, 60 microliters of ovomucoid solution, and 520 microliters of EBSS. After the incubation, transfer the papain solution containing the partially digested retinas to reaction tube 2, and then triturate the retinas 10 times with a fire-polished pipette. Gently layer the single-cell suspension on top of the ovomucoid solution in reaction tube 1, and centrifuge the cell solution for 5 minutes at 300 Gs and room temperature. Then discard the supernatant and resuspend the cells in 500 microliters of MACS buffer. To sort by MACS, first incubate the cells in 10 micrograms per milliliter of rat anti-CD73 antibody on ice. After 5 minutes, wash the cells in 10 milliliters of MACS buffer. Then resuspend the pellet in 480 microliters of MACS buffer and incubate the cells in 120 microliters of Go anti-rat IgG micro-beads on ice without agitation, shaking, or mixing. After 15 minutes, wash the cells in 5 milliliters of MACS buffer. During the wash, place an LS column onto a MACS magnet and put a pre-separation filter on top of the column. Next, hydrate the filter and the column with 3 milliliters of MACS buffer, collecting the buffer in a 15 milliliter tube labeled W for wash. When the cells have finished spinning down, resuspend the pellet in 500 microliters of MACS buffer and then load the cells into the filter. Add 1 milliliter of MACS buffer on top of the cells, and then collect the negative fraction in a 15 milliliter reaction tube labeled minus for negative. Next, add 3 milliliters of MACS buffer to the column 3 times to wash it. Now remove the column from the magnetic stand, install it on top of a 15 milliliter reaction tube labeled plus for positive, and quickly load 5 milliliters of MACS buffer into the column. Plunge all of the buffer through the column, and then, after spinning down the positive fraction, resuspend the pellet in 500 microliters of MACS buffer. Place the cells at 4 degrees Celsius and count them. Then adjust the concentration to 2 times 10 to the 5th cells per microliter in MACS buffer, keeping the cells at 4 degrees Celsius. For transplantation of the isolated photoreceptor precursor cells, dilate the pupils of a sedated adult mouse with a drop of phenylephrine tropicamide. While the pupils dilate, use a diamond pen to cut a cover slide into approximately 5 by 5 mm pieces. Place the mouse under a stereo microscope, restrain the animal in a mouse head holder, and then apply a drop of Vidicic gel to each eye to prevent drying. Now use a 30-gauge half-inch needle to make a small hole at the border between the sclera and the cornea. Then place one of the 5 by 5 mm cover slide pieces on top of the cornea, allowing direct visualization of the retina. Next, flush a pre-sterilized microliter syringe several times with sterile deionized water, and then load the syringe with 1 microliter of the sorted cell suspension. Direct the needle diagonally through the conjunctiva and sclera, placing it in the nasal half of the retina. Then gently punch a hole through the retina to the subretinal space and inject the cells. Confirm the bullous uniform detachment of the retina, which should cover approximately a quarter of the retinal space without any bleeding. Then withdraw the syringe gently and flush it several more times with deionized water. Finally, release the mouse from the restrainer, inject the animal with adipamazole hydrochloride to reverse the effects of the anesthesia, and allow it to recover in a dark 25 degrees Celsius chamber. In order to assess the ability of rod photoreceptors to integrate into the mouse retina, a mouse reporter line was used in which GFP is driven by the neural retina leucine zipper, or NRL, promoter. As demonstrated in these histograms, the initial retinal cell suspension contained 30.4% GFP-positive rod photoreceptors. Following CD73-based MACS sorting, an enrichment of up to 86.9% of the GFP-positive photoreceptor precursor cells was observed in the CD73-positive fraction. In the CD73-negative fraction, only 9.9% of the cells were positive for GFP. The enrichment of NRL GFP-positive cells following CD73-based MACS is also observed after plating in vitro. The donor cells remain at the site of injection, as defined by the bullous detached host retina, and integrate into the outer nuclear layer of the host retina, acquiring the morphology of mature photoreceptors.
Once mastered, magnetic associated cell sorting can be performed within 2 hours if correctly done.
This procedure can be also adapted for the enrichment of stem cell-derived photoreceptors or even other retinal stem cells, given the availability of specific antibodies, to answer additional questions in regard to restorative or rescue by cell transplantation for the retina.
After watching this video, you should have a good understanding of how to transplant MACS-enriched photoreceptor precursor cells to the subretinal space of recipient mice.